I know that most of you do not know much about glycans, so I'll make a small introduction. So the protein you see revolving in the middle, that's the S protein of the SARS-CoV-2 virus, and everything hairy you see on the surface are the glycans. So we cannot understand the function of this protein if we are ignoring the glycans. So the best analogy I can make is this bird on the left, where you have a bird without a single feather. This bird is not a functional bird, and protein without its glycans is just like a bird without its feathers. It cannot perform all functions, because proteins, glycans can change the structure of a protein. If you change a structure of a glycans attached to immunoglobulin, it will convert it from pro-inflammatory to anti-inflammatory molecule. So if you are studying only polypeptide part, and we use this bird as an analogy, if you try to understand flight, and you use a birdless, featherless bird as a model, this will be the closest to the flight you can come. Just bird walking around, because bird without its feathers cannot fly. Protein without its glycans cannot perform full functions. Glycans give the full complexity of a protein structure. They explain protein function. And just a word of caution at the beginning, this has nothing to do with the glycation. Glycation is a random damage. It's a chemical reaction between glucose and the amino acids. We quantify it as HbA1c, advanced glycation end products. This is something completely different. Glycation, random damage. Glycosylation, sophisticated modification, which makes proteins way more elaborate. Actually, glycosylation adds additional layer of complexity between gene sequence and a protein function. So polypeptide part, this is so bacteria. You have a gene, you have a protein, you have a function. This is the way how bacteria function. And E. coli has 5,000 genes. We have 20,000 genes. We are not only 5 times more complicated than an E. coli. So at one point in evolution, when we were becoming multicellular, we invented glycosylation. We invented a network interactions of dozens of proteins which work together to make a glycan structure, which then becomes a part of a protein. An alternative glycosylation, attaching different glycans to the same glycosylation site, is analogous to coding mutation. It's changing the protein function, but it's being inherited as a complex trait. And this is something we really clearly demonstrated when we looked at the study of cohorts of over 100 mice strains generated as a collaborative cross cohort. So they started from the 8 founder strains, crossed them for 3 generations, and then inbred the cousins into strains. And some of these cousins, some of these strains became structurally and functionally distinct from the rest of the population without a single mutation. They became different just because they reshuffled alleles in this complex network which glycosylates proteins. And we see the same in humans. We are different. On the right-hand side of this panel, it's just 6 random individuals, different color, different glycan, IgG glycan composition. People are different. Big part of inter-individual variation, so what makes us different from another human, are glycans. Actually, blood groups are also glycans. We just don't think it in that way. So glycans are the molecules which can help us understand why are people different. So why people don't look glycans more? Why didn't you learn about glycans during your studies? And the answer is because glycans are extremely complicated. You can have many different structures attached to the same glycosylation site. You can have multiple glycosylation sites on the same protein, and it really takes many chemical steps to analyze a glycan, especially in a high-throughput manner. So many people just stay away of it. They don't do it, and they do their science on this featherless bird, and they think they're doing a high-quality science. They could be the best scientists in the world with the best design experiment and execute without any flaw, but they would never understand flight, because bird without its feathers cannot fly, protein without its glycans does not function normally. We were the first who went into the field of high-throughput glycomics. 15 years ago, we studied the first cohort of 1,000 people. So far, we analyzed over 200,000 people. Approximately 80% of the global published glycoms came from my lab. And we did it in collaboration with some of the leading researchers all around the world. And this enabled us to shift glycomics into the, more or less into the mainstream. At least now we are able to publish glycomic studies in a reasonably decent journals, including some of the leading journals in the field. And the first question we asked is, how are genes linked to glycans? So how genes regulate glycosylation? And through a series of GWAS studies we published in nearly 15 years now, we mapped very large network of genes which regulate glycosylation. For example, immunoglobulin G has more or less 4 key enzymes which generate glycans attached to IgG, but there are over 40 regulatory proteins. So the regulatory network is way, way larger than just the production. So the glycosyl transferases, these are the blue-collar workers. All the white collars are the other mostly unknown proteins at the moment. But these genes are highly pleiotropic for a number of different diseases. So we did the pleiotropy study and identified nearly 100 different diseases and conditions which associate with genes which are involved in IgG glycosylation on a SNP level. And this is highly protein specific. So we did the parallel GWAS of IgG and a transferrin, same 1,000 people, nearly the same glycan structures, but attached to 2 different proteins. Enzymes which make them are the same, but the regulatory network was completely different. So different proteins were regulating glycosylation, the synthesis of the same glycan in a different protein. And even when it was the same gene, for example, the enzyme which adds fucose, the FUT8, it was a different SNPs in a gene which were regulatory in transferrin and IgG because it was produced in a different tissue under the regulatory network of different transcription factors. So this all created a very complex process which peaked in evolution at the moment when we were becoming multicellular. So when we were developing things like the tissues, like the initial organs, the interactions between cells, new proteins were evolving. They got some specific functions and they kept them for the rest of evolution. This is still unpublished, but it should be published soon. So the hypothesis which Vlatko and I published over 10 years ago, so are glycans the third revolution in evolution? Yes, glycans are the revolution in evolution which enable the multicellular life. So to summarize this introduction, glycans are the molecular effectors. They are part of the protein molecule. They integrate genetic information, but also epigenetic information, and the direct environmental factors into chemical structures which do the work. So we have all the evolutionary wisdom. This is what genes, what is encoded in genes. We have all the wisdom for maybe last 2 generations, which is encoded in epigenetics. Then we have our lifestyle, diet, stress, whatever we do, integrating into the molecules, which especially in the immune system are one of the key regulators. So glycans attached to IgG are one of the key functional effectors which balance inflammation on multiple levels, from binding to the different receptors to affecting different cells in the immune system. And this changes a lot with age. So as we are younger, we mostly have more of the glycans which suppress inflammation. And when we are older, we have more glycans which promote inflammation. And when we looked in populations, and this is 5 different populations, over 1,000 people in each of them, we see that these glycans change a lot with age. And this realization that the glycans change with age was the basis of the GlycanAge test we developed a decade ago, which we now know is a functional effector of inflammation, which can be modified by lifestyle, which can be modified by pharmacotherapy. It can be easily quantified, measured, interpreted. And of course, I'm of course conflicted here. I am the inventor of the test. I am a co-founder of a company. So I'll try just to present the data, and you make your own conclusions. But being involved in the company also enabled me to bring company here, so it's in front of the door. They're giving a special discount, so if you're interested, talk to them. We have epigenetics, which we heard about in the last presentation, and we have a glycans. So the key difference between epigenetics and glycans is that epigenetics is an information. You look at it, but you don't really know what is the functional relevance. We have to do a lot of studies to understand what each epigenetic CpG site actually does. While glycans are the functional effectors. There is a lot of knowledge what each of these molecules is doing and what will happen if you change this specific molecule. Also, when a large study was done on Weill Cornell in Qatar, they quantified over 6,000 different molecules, more or less everything what can be measured in a human, from proteomics, metabolomics, glycomics, genomics, epigenomics. And out of 6,304 molecules which they quantified, in the top 20, which correlated with age, 9 of them were glycans, immunoglobulin glycans. The others were mostly steroid hormones. So glycans definitely strongly correlate with age. And there are literally hundreds of papers, both from our lab and from other labs, showing how glycans change in the different diseases. So glycans definitely change a lot in different diseases. This excellent review from last year, it's freely open. So if you're interested, you can have a look. When we looked around the world, we collected 27 populations, 100 individuals in each, from all parts of the world. We realized that people in different parts of the world are different. So when you look at their glycans on IgG, they're different. But when we tried to find what is actually the main confounder, we were quite surprised to see that it's actually expected lifespan and the Human Development Index. So people living in the countries where the expected lifespan is short, and these are usually the poor countries, have glycans moving much faster toward these old glycans than people living in wealthy countries. Approximately 40% of that is genetic. So heritability of the GlycanAge clock is approximately 40%. 60% is a lifestyle. So lifestyle is dominating the pace of changes in our glycans. And this acceleration is usually linked to unhealthy life habits. Also, it correlates with unhealthy biomarkers. But there are different kinds of acceleration. For example, when we looked in a people with a Down syndrome, in Down syndrome, we see a very clear acceleration of a glycan aging. But this acceleration happens early in life. So there is a burst of acceleration, and then the pace of aging more or less is the same as in disomic individuals. And in a paper we recently published in eBiomedicine, we identify one kinase, the DARK1A, which is linked to the unrepaired DNA damage to be likely causative for this. And then if this is really true, it's logical that we have the acceleration in the young age, because then there's much more DNA division than we have it in the old age. When we looked for correlation, actually, this is not our work. When a team in Edinburgh looked at correlation between the different aging clocks, the acceleration between the glycan aging and epigenetic aging was actually very low, at least for these epigenetic clocks. So one has to be careful when we talk about epigenetic aging. As Steve emphasized, different epigenetic clocks are different. They measure different things. They only have the common name, but they have nothing in common. So you cannot just say epigenetic clock. You have to say exactly which epigenetic clock. So with these epigenetic clocks which they tested, acceleration in glycan and epigenetic aging was different. So obviously, these were quantifying different aspects of aging. When we looked in patient cohorts, and we did dozens of different diseases, in the majority of diseases which have low-grade chronic inflammation as one of the elements in the disease, we did see accelerated aging. So the last 4 bars on the right are just young and old people in the population cohort, and all other bars are different diseases. So we clearly see that people with many diseases have GlycanAge acceleration, and often this happens up to a decade before the disease is actually diagnosed. So what we believe that we see here is that we see the mechanistic role of IgG glycome, which is promoting or suppressing inflammation, and in this way contributing to the lower or higher level of chronic systemic inflammation, which then contributes to the disease, which will then again be dependent on a genetic, epigenetic, and environmental factors, whether you will get a Crohn or arthritis or lupus or whatever. I will show you one example, because I don't have time to go into all of them, where I think that we have relatively solidly looked at all aspects to try to understand what is going on, and this is the cardiovascular diseases. So initially, we did a large collaborative study with Tim and Jim Wilson from Edinburgh, where we looked at over 3,500 people, just looked at the glycans today and the cardiovascular disease risk score, and we saw a correlation. Correlation which was replicating, suggesting these two things are somehow linked. Then we did a number of longitudinal studies, including the 2,000 twins from the TIMS cohort, which we analyzed in 3 time points across 15 years, showing that the change in IgG glycome comes before the hypertension develops. So it actually predicts future hypertension. Then we managed to persuade owners of the EPI cohort, and the EPI cohort was collected over 30 years ago, 300,000 in Europe. We got access to the 27,000 people from the German subset. There was a clinical follow-up, so we had a sample, and we knew who will and who will not develop a cardiovascular event, meaning a heart attack or stroke. And then we looked at predictive power, and the glycans in men were as predictive as the entire AHA score, and in women, a single glycan was more predictive than the AHA score for the future cardiovascular events. But this is one paper, one has to replicate. We did another large cohort, and again, we see a highly stratified men-woman biomarkers, meaning one glycans predict heart attack and stroke in women, the other predicts in men, and the hazard ratios here are reasonable. You know, this is not a minor effect. It's a significant prediction of heart attack and stroke. And then we did additional 2 replications, this time with the colleagues from Harvard in 2 large prospective cohorts. And this is the basis of the future CardioAge test, which is going to be commercialized through GlycanAge, which we believe, at least for now, we are confident in women, will be highly predictive of the heart attack and stroke in the future. And we are trying to understand why are these glycans linked to hypertension, why are they linked to stroke. And there is a molecular mechanism, while IgG is being recycled in endothelial cells, it somehow affects both the nitric oxide release and not linked to hypertension, but linked to diabetes, also the insulin resistance. So it seems that these are mouse experiments. So if you put mouse on a high-fat diet, it's losing the IgG cellulation, and this hypocellated IgG is promoting both insulin resistance and hypertension. And there is a dietary supplement called MANAC, which is N-acetylmanosamine, which is a precursor for salicylic acid. If you supplement mouse with this MANAC, you can save the cellulation of IgG. So the mice will, of course, become obese because you give them high-fat diet, a little bit of sugar on top of that. But they will not lose IgG cellulation, and they will not develop hypertension. So for mice, there's a magic pill how you can be fat and do not develop hypertension. Whether it works in humans or not, we have no ideas. But this is the direction we want to go for these immunoglobulin, glycans, and hypertension. In the diabetes part of the story, we have also very interesting result. Here, these are not the immunoglobulin glycans. These are the total plasma glycans, which do not come from B cells. They come from liver. And what we see here, and this is again these 2,000 twins from the TIMS cohort, is that as somebody is approaching diabetes or insulin resistance, glycans are moving from the healthy state toward this, call it the diabetes state. So again, problem with diabetes is that diabetes is not a disease. Diabetes is a symptom, which has many different molecular pathways which can lead to diabetes. And we believe that if for one of them, glycans are highly important. And this is something we did actually a long time ago, where we looked at the people who develop hyperglycemia in acute disease. So we're working with the ICU, people accepted to ICU, hyperglycemic, normal glycemic. And then we went to them 2 months later when they fully recovered and used glycans to predict who will and who will not develop hyperglycemia. And we had AUC something like 0.97 or something like that. Nearly perfect prediction of who will and who will not develop hyperglycemia when put under stress of acute infection in most cases. And these very same glycans, which are predictive of hyperglycemia, are also predicting the spikes in lipids and also largely in glucose in postprandial in this PREDICT study we did together with Tim. So glycans, which are predictors of a disease, are predicting spikes in response to food. So most probably they are regulating the half-life of all the receptors on a membrane. And the great thing about it is that if you change your diet, you can improve these glycans. So healthy lifestyle will move these glycans in the opposite direction from disease progression. And we know that from the initial paper on this progression to diabetes, this is not 0-1 transition, it's a continuous process from health to disease, which you can revert with a dietary intervention. So kind of to summarize this part of my talk, the idea is that the transition from health to a disease is not 1-0 process. It's a continuous process which can go for a decade or more, where first we are losing some kind of a molecular response, our body is responding, we are adapting, we are compensating, until we really feel pain somewhere and something does not function in our organism. And usually we go to hospital when we feel pain. But this is way beyond the point of no return. And most of these people will get a diagnosis, will become diagnosed of a chronic disease, and they become a lifelong customers of the pharma industry, where they will be drugged and drugged and drugged forever, because most of the modern medicines is not dealing with the disease, but with the symptoms. So we are mostly treating symptoms and not the cause of a disease. If we can identify people on this trajectory early, if we can identify biomarkers, we change much before there is a disease. And if there is a way to revert them, maybe we can postpone disease, or maybe even completely prevent it. And I believe that glycans are great there. I'm very happy to see that the consortium on biomarkers of aging, in the paper they published very recently, clearly identified glycans as the only biomarker of age which is currently being used to monitor lifestyle interventions and which is predictive of multiple diseases. So, what can we do? What can we do to improve our glycan biomarkers, slow down, or even revert the glycan clock of aging? So we know that there are at least 16 genetic loci which are involved in the regulation of GlycanAge clock of aging. I will not talk much about it because Vlatka has a presentation just even after the coffee break. She'll tell you many more details about the work we do together about the epigenetic regulation on these genes. We did a number of studies with the different really aggressive therapies, different biologicals, anti-inflammatory biologicals, which would improve IgG glycosylation and suppress inflammation in the majority of people. But of course, you do not want to use infliximab as a kind of a preventive anti-aging drug. So there will be a special issue of the expert opinion on therapeutic targets about therapeutic targets for longevity. I wrote a paper about glycans and how we can use, in which way we could use this large network of genes which regulate glycosylation to potentially address inflammation also preventively. It will be published in a few weeks. You could have a look. Another thing we know that menopause strongly associates with extensive changes in IgG glycome. So when we look at the trajectory of changes in the GlycanAge in men and women, men are mostly aging linearly. And women are mostly protected from this aging during the most of their productive age, and there is extensive acceleration, and then they keep up with men later. And these are 4 different cohorts, so these things clearly replicate. And on an individual level, this can be really rapid. Again, we looked at this cohort of 2,000 twins together with them, where we had them in 3 time points, and we see that this period of transition, there is a very large acceleration of GlycanAge, and we believe that GlycanAge can actually be very informative here, because the problem in the perimenopause period is that hormones are not really informative, because they fluctuate too much. You kind of have to accumulate 30-day urine to have a reliable readout, while the immunoglobulin glycans are strongly affected by estrogen, and they have a half-life of 3 weeks. So by measuring immunoglobulin glycans today, you're practically integrating estrogen concentration for a couple of weeks. So IgG glycome today is something like HbA1c for glucose. It's telling you the average load of estrogen for the last couple of weeks. If you do a bariatric surgery, your glycome would improve, nearly everybody. Oh, sorry, again, disclaimer, we filed a patent for that. I have commercial interest in the aspect of application of GlycanAge in the menopause. If you do bariatric surgery, your GlycanAge would improve in the majority of people. We had a patient improving for 35, 37 years in 6 months. Not everybody will improve that much, and majority of you do not want to do bariatric surgery, which is okay. Why would we go for surgery to lose weight? So we looked whether regular weight loss is also effective in reducing GlycanAge. Again, on the same cohort of 2,000 twins in 3 time points, we had twins who were gaining weight and twins who were losing weight between the time points. And in all periods of life, in the 30s, in the 40s, in the 50s, if you are gaining weight, you have accelerated glycanaging. If you are losing weight, you are decelerating glycanaging. So it doesn't matter when, weight loss is beneficial. And we did, together with a number of colleagues of Europe, in Europe, a large Diogenes study where they had 1,000 people on 800 calories for 2 months. That's the equivalent of torture. You are kind of torturing people with 800-calorie diet, and the GlycanAge improved in most of them, but nobody can keep a low-calorie diet. That's not an idea. But the idea was, let's see whether there is a good diet which will maintain these beneficial effects. So on average, these people lost 10 kilos in the first 2 months. Then they were given advices for several different diets, including the high glucose, high protein, low glucose, low protein, all different magic diets. And each of these diets, we had some people increasing GlycanAge, some people decreasing GlycanAge. And not a tiny little bit, there were significant effects. And I think the conclusion here is very clear and exactly the same what Tim said in his lecture. There is no standard human. There is no one-size-fits-all approach to healthy diet. This is why we have thousands of books about healthy diet, because every diet will be good for somebody. There is no diet which will be good for everybody. And we have to have biomarkers to see which diet works, measure it before, measure it later, and see whether this diet is good for me or not. The same goes for physical activity. So when we started the GlycanAge, the first idea was that we'll work with the gyms, people will exercise. After exercising, they will get younger. And some of our colleagues from the different kinesiology universities and so on, they were really excited. And we had thousands of people exercising. And in the majority of them, we actually see the increase in GlycanAging. So when you take a middle-aged couch potato, force them to exercise, push them harder, you do them damage. Exercise has to be carefully optimized. It has to be matched with relaxation. And too much exercise doesn't help. We published a number of papers, we'll publish a number more. So it's physical activity has to be carefully tuned to an individual. Microbiome is extremely important, as you have heard from Tim. We did a number of studies with the microbiome transplantation, where you take a healthy microbiome from one person, put it in another person, and their glycans would improve. We are doing some of the studies together with Tim to see whether this microbe-based diet will also improve the glycome. Some of the clinics which do anti-aging are actually quite successful. There are some clinics where nearly all patients which go into the clinic become 15, 20, 30 years younger within a year or so. Some individuals are highly successful. Tim showed his story. What he didn't show that he actually recorded the Instagram Live about what he did. So you can look at this Instagram Live to see the trick, how he managed to lose 11 GlycanAges within half of the year. And to finalize my story, I would like to show you this tire on the right-hand side of the panel. So imagine if we would change tires on our cars only after they explode on the highway. Driving would be deadly, not because of us having old tires, but because of other people having old tires. And we don't do it. We do a lot of preventive maintenance on our cars. We change tires, we change oil, we go for the regular maintenance, but we do not do it for our body. We keep our body running until it breaks down. Only after something breaks down, then we try to fix it. Tim was happy. He had a minor stroke, or what it was, and then he changed his approach. I never had a stroke, so I still do it the way I did before. But maybe we should be smarter and somehow do this preventive medicine, personalized preventive medicine, and prevent diseases. And I really think that the glycan biomarkers can here be really helpful, because the key problem is, I know what is healthy. I should eat less. Definitely I should eat less. And probably better, but also less, because I eat too much. But I don't do it. I don't do it because, the reward will come in a decade, 2 decades. I'm not going to skip that cake in a coffee break because of something what will happen in 20 years. It's unknown. But if I have a biomarker which I can measure, and which tells me, you're getting better, you're getting worse, my motivation could be better. And we do see that. This thing here, the smartwatch, it can actually motivate me to stand up and walk and say, you have to stand up and walk for 2 minutes because you will lose your star for standing up 12 hours a day or whatever. So this feedback, we need the feedback. GlycanAge can give us feedback and can also tell us that maybe we are doing something wrong. And we have, since GlycanAge is being available in, I think, over 1,000 clinics all around the world, there's a lot of feedback of people improving, learning, and becoming more healthy because they use it. So the idea is, we have the objective insight into current state of low-grade inflammation. We have a tool to quantify it, and it gives us objective feedback. And the GlycanAge is just the beginning. The glycans are the third revolution evolution. We are opening the whole new area of glycans. We are about to launch the MetaAge, the CardioAge, and the MenoAge, which are still be just the tip of the iceberg. I have to acknowledge that I have a very large, fantastic team of people, many of them with me for more than 15 years now, and they do all the work. I'm also just travel around and talk about it. And we burn huge amounts of money. We got over 40 million of different, mostly European grants, to analyze all these glycans and to accumulate all this data which we have now. And I thank you all for your attention, and I'll be happy to take questions.